तो अब हम बात करेंगे पैथोजेनेसिस ऑफ मास्थीनिया ग्रेविस. अभी तक वी ऑलरेडी डिस्कस अबाउट द normal function of the neuromuscular junction, that how it is work, right? So like we have to make sorry. So for example, this is a spinal cord, right? And there is a cell body and the nerve is coming out and is spanning, right? And we have sarcolemma crest where we have acetylcholine receptors, and we have two type receptors here, that is sodium channel and calcium voltage gated channels, right? And of course we have these acetylcholine receptors which is present on the crest of the sarcolemma, right, where the acetylcholine has been attached and the local action potential is generated. Then obviously for that local action potential has been transmitted to the full length of the muscle, and the contraction of a muscle is, as we all know, it is a disease of autoimmune disorder, in which disease in which there's so many autoantibodies is developed. So in Myasthenia gravis we have two types of antibodies, right? So auto antibody, or we can say antibody, is developed, right? So in Myasthenia gravis we have two types of antibody which is developed. Number one, which is 85%, that is the antibody developed against the acetylcholine receptors. So the antibody is developed against the acetylcholine receptor which is present on the crest of the sarcolemma, acetylcholine receptors. On the other hand, so this is the most common form of antibody which is developed in that particular disease, and it is about 85%. So, and the second one is that hope you remember there is another protein which we already discussed, that is also present on the sarcolemma, and the regulation of this musk, the function of this musk protein, what is the function of this musk protein? That it regulate the density of these acetylcholine receptors. So sometime what happens, these antibodies develop against this. So the antibody is developed against musk protein, right? These antibody are targeting these musk protein. So you can easily understand that if the receptors has been targeted by the antibody, what happens, that these are less in numbers. Same if it is spared and autoantibody is not developed against these receptor, but it has been developed against these musk protein, and you know that the function of musk protein is very important. It regulates the density or the concentration of this. So obviously we do not have enough receptors to get acetylcholine attached, and the action potential has been generated. That is why the disease is confirmed. That what is the prevalence and in which age group we normally found this disease, right? So if we make a graph, so it gives us a biphasic graph, right? Biphasic graph, what does it mean? So initially, if you say the 20 to 30 age group, most commonly this disease will be developed or find out in the female, right? So initial means that the younger females are usually frequently involved or develop that particular disease. But as we getting older, that is if we can say 40 to 60 age group, so male are dominant, right? Male are dominant as far as the myasthenia gravis. Now the question is that what is the reason behind it? It is because of hyperplasia, usually find out in the hyperplasia of, it is due to hyperplasia of thymus gland. So the reason being the female is more involved. इसमें ये ज़्यादा क्यों इन्वॉल्व होते हैं यंगर फीमेल? वो इसलिए होती हैं क्योंकि जो हाइपरप्लेजिया ऑफ थाइमस ग्लैंड होता है वो कॉमन है, वो फ्रिक्वेंट है इन यंगर फीमेल्स, राइट? बट ऑन द अदर हैंड व्हेन वी आर गेटिंग ओल्डर, जैसे हम बूढ़े होते हैं या हम ओल्ड होने लगते हैं, तो जो जो ज़्यादा थायोमोमा होता है, थायोमोमा, so the rate or the prevalence of thymoma is more in the male group. So that is why there is a good chance that male will be develop that particular disease in the later half of the disease. So now I hope you understand the prevalence, why it is common in the male, in younger female, and why it is common in older males, right? Now coming to the pathogenesis, exact pathogenesis of myasthenia gravis, what actually happens. So as we already know that more than 85%, there is a autoantibody generate against develop against acetylcholine receptors, right? This is a fact now, everybody knows about it. And which type of antibody? IgG, right? So which type of antibody is developed? That is IgG. So IgG is the type of antibody, right? If you see, this is the structure of antibody, right? Okay, so it is the type of antibody which can be passed through the placenta. I think you remember that particular point. So another very characteristic of IgG is that it is bivalent. What does it mean, bivalent? Bivalent means that it can be attached to two allergens, right? Simultaneously, right? Okay, means now we have to see that how these antibody is targeting these receptors. So if we अगर हम इस डायग्राम को बड़ा करके देखें तो हम इसको आसानी से समझ सकते हैं, ठीक है? तो फॉर एग्जांपल दिस कोलिन रिसेप्टर्स, राइट? दीज आर नथिंग बट what they are acetylcholine and receptor. So these are acetylcholine receptors. Now there is a antibody which we just discussed, and it is a IgG, and the characteristic of IgG is that it can be connect, or it can be bind with two allergens, right? That is why it is called as bivalent, right? I hope you can see this diagram, that this is the antibody, this is the first antibody, and this is the second antibody, and these are the three. You can imagine like this also, right? Okay, and we can make another receptor here. Okay, so what we can see in this diagram, that one antibody is attached to two receptors like this. So what it does, what happens actually after this clustering, so there is a cluster formation, pathological cluster formation occur. तो अगर हम ये देखेंगे कि इसमें क्या होता है कि जो रिसेप्टर्स होते हैं उस पर जब एंटीबॉडी आके अटैच होती है, तो क्योंकि ये बाइवेलेंट एंटीबॉडी होती है, तो एक एंटीबॉडी दो रिसेप्टर से अटैच हो जाती है, एंड दिस इज हाउ सो मेनी एंटीबॉडी अटैच विद द दीज रिसेप्टर, एसिटाइल कोलिन रिसेप्टर। उसके नतीजे में क्या बन जाता है, इसके नतीजे में क्लस्टर फॉर्मेशन विच इज अ पैथोलॉजिकल इन नेचर बन जाता है। नाउ इट इज नॉट अप टू दिस लेवल। इसके बाद क्या होता है, द सेल you know sense that there is some pathology occur इन द सेल। सो क्या करता है सेल, जो मसल जो सेल होता है वो इसको करेक्ट करना चाहता है, एंड इट इज करेक्टेड बाय। सो अगर हम इस पार्ट को यहाँ देखें, सो फॉर एग्जांपल these are the antibodies, right, which is attached to these. So what happens, this cell is senses that there's something wrong occurs in that particular structure or the receptor, and it moves inward, right, it move inward by the process of endocytosis. So it can be replaced, right. So उसके बाद क्या होता है, they wants to go inside, right, by the sensing of that particular cell that there's some kind of abnormality occur, and they move inside like this. They move inside, and these are the receptors, we can say, with the antibodies, right, and ultimately what happens, it closes off, right, and they will form another membrane upon this, and this is closed like this, and we have a cluster of antibody and acetylcholine receptors like this inside, and we have antibodies with it, obviously. Now when it is reaches inside the cell, the next step that it should be destroyed. It should be destroyed with the help of lysozyme. Okay, now the point is, you just focus on this particular point. When these receptors are going inside, okay, and it has been destroyed, so obviously it should be replaced by the new ones. Okay, and who is responsible for this? This guy is responsible to replace or maintain the density or the concentration of these acetylcholine receptors. But the problem is that we have so many antibodies and this pathological cluster formation, and this process is going inside these cluster formation, going inside into the cell, and it has been destroyed. तो उससे क्या होता है कि ये जो प्रोसेस होता है, that process of engulfing, this process of engulfing cluster is very fast. It's rapid actually. It's very fast, or we can say these are rapid. This process is rapid, right? That protein is unable to replace them. समझ में आई बात? मतलब कि ये जितनी तेजी से इसका डिस्ट्रक्शन हो रहा है, उतनी ही तेजी से इसको रिप्लेस करना चाहिए these receptors. Otherwise there is no receptor. So हमारे पास कितना भी एसिटाइल कोलिन हो, अगर उसका रिसेप्टर मौजूद नहीं है तो वो वर्क नहीं कर सकता. तो उसका रिस्पॉन्सिबल होता है मस्क प्रोटीन. और ये जो प्रोसेस होता है engulfing and destroying these cluster, it is rapid in such a manner that musk protein cannot replace these clusters, and that is why the diseases develop, or we can say the symptom will arises, right? So this is the one part of the pathogenesis according to the antibody targeting these receptors. The second part of this antibody is that this IgG these IgG antibody has another function. वो क्या होता है? So another function is that it stimulate the complement system. ये क्या करते हैं? ये complement system को एक्टिवेट कर देते हैं. स्टिमुलेट और एक्टिवेट, नॉट it activate the complement system. Which one? IgG, which is a primary antibody in Myasthenia gravis. So what happens when this is activated, this complement system is activated, it is further destroying the muscle cell. But how? We'll look after right now. So for example, this is the choline receptor and we have antibody attached to the receptor, unfortunately, right? So what another function is that it stimulates the complement system, complement or activate the complement system, right? How it stimulate? It stimulate complement system 1, right? Then this complement system 1 is activated complement system 4, right? And this 4 is stimulated or activated C2. C2 is activated C3B, right? This activated C5B, okay? Then 6, C6, then C7, then C8, and then C9. So you can, I hope you can easily understand, and this is the series, this is the correct series, right? That it activate first complement system 1, complement 1, then 4, then 2, then 3B, then 5B, 6, 7, 8 and 9, and ultimately when these all are activated, so the function of this activation, complement system activation, what it does, it makes one channel inside these molecules. We can say like it makes a pore in the cell membrane. This is the cell membrane, you can easily see this is the cell membrane. So when these are activated, what happens, it makes like a tunnel, right? They form like structure like a tunnel in cell membrane, right? So आप अब आसानी से इस बात को समझ सकते हैं कि अगर किसी भी तरह से सेल मेम्ब्रेन को डैमेज कर दे तो ऑब्वियसली इट इज डैमेज्ड, राइट? It is damaged, right? So when it is damaged, so all the material which should not be going inside, that is extracellular material, can go easily inside, right? And those molecules, those enzyme, those cell organelles which should not be go outside, it can go in the extracellular space, and what ultimately happens, obviously cell will be die, cell death, right? Cell death obviously occur. So these are the two important process of the pathogenesis of antibody which is working inside. That is, number one, it form the pathological clustering, and number two, it activate the complementary system, right? I hope you understand that how it works, right? Now we have to see that what are the changes occur after these two pathological process occur in. So you can write number one. Obviously, if this processes occur multiple times, so what happens? The number of crests will be more or decreases less. Yes, you're right. The number of crest will decreases. These are the crest, right? These are the crest, right? These are the crest. So you can see that one crest is over. These are the crest. Okay. Number two pathological thing what we can see that it increases the distance, right? Distance between the synaptic cleft, or we can say between the neuron presynaptic neuron and sarcolemma. So increase. Okay. And the third and important point is that when we do not have these crests, तो क्या होगा? इसका surface area बढ़ जाएगा या कम हो जाएगा? Yes, decreases the surface area of sarcolemma or muscle cell membrane, whatever you can see. Cell membrane, right? Again we can repeat whatever happens in our body voluntary, it will coming from the cortex, then spinal cord, and the action potential is coming from the motor nerve up to the muscles, which is known as neuromuscular junction. So that action potential is reaches to the sodium ion channel, then sodium influx occur, so many cations is there, and the action potential or generated, which is open the calcium voltage gated channel, which is essential for vacuoles attached to the neurolemma in a presynaptic neuron. When calcium is going inside, it opens the site where these vacuoles and the membrane can easily bind to each other and exocytosis occur, and we have acetylcholine in the neuronal cleft. Okay, when it is the acetylcholine is coming, these attach with the receptors, and when these acetylcholine is attached to receptor, it opens and it influx so many cation inside the cell and generated local action potential, and which ultimately convert into the action potential for the muscle and contraction occur. Myasthenia gravis is an autoimmune disorder, and it is having two types of antibody. That is, it can be developed against the acetylcholine receptor, which is the most common form, and sometimes the antibody is developed against the MuSK protein, which is the function of MuSK protein is to maintain the concentration of these acetylcholine receptor in the cell membrane or sarcolemma. Okay, and the pathogenesis is that it is the antibody which is generated is IgG type, which is bivalent, and it attaches two receptor at a time and makes the pathological clustering, which is very which makes these receptor malfunction, and cell senses this malfunction, और उसके साथ क्या होता है, it is going inside into the with the process of endocytosis and killed by the lysozyme. But this process is very rapid, so that in that manner that MuSK protein cannot replace in time, and the degeneration is more and replacement is less. Because of this, the disease is developed or the clinical feature is coming, right? The second thing which happens because of the antibody production, that it also activate complement system. How it activate? First is activate this complement system 1, which activate 4, 2, 3b, 5b, 6, 7, 8, 9. And what ultimately these complement system do, that that it makes a pore and damage the cell membrane, which is lethal to any cell, right? If you do anything to the cell membrane, it will ultimately giving death to that particular cell. So this is all about myasthenia gravis pathogenesis. Now we will move on towards the clinical features of myasthenia gravis. The hallmark of neuromuscular junction disease is that it produces painless muscle weakness. So obviously the most important clinical feature in case of myasthenia gravis, clinical feature. So the most important clinical feature in this case, obviously, it is painless muscle weakness. The second point in myasthenia gravis, that usually frequently that disease is start from the head and going towards the trunk, right? So obviously it involves cranial muscles first, or we can say it affect cranial nerves, is especially extraocular, which supply the extraocular muscles. So because of this, what we have most important is we have ptosis. We can have difficulty in speech, right? Difficulty in speech, or we can say this is dysarthria, right? We can have difficulty in swallowing, right? And we can also another very important clinical feature of the myasthenia gravis, that they are fluctuating, that weakness is fluctuating in nature. फ्लक्चुएटिंग इन नेचर मतलब कि ये कभी तो वीकनेस ज़्यादा हो जाएगी और कभी नॉर्मल रहेगी, राइट? मसल वीकनेस, दिस इज द नेचर ऑफ द डिसऑर्डर.